be doing this twice. Okay, so I would call this chapter 8, part 2. Right? Hopefully we won't have to have a part 3. Hopefully this will be enough. Again, brief review. It should be a review for you, but I am going to go into a little about the same amount of detail I usually do in Bio 1. And the reason for that is because in the lab, most of the activities that we're going to do starting next week are going to be about biochemistry. They're going to be about how bacteria do their metabolism. So it is important that we remember this stuff a little bit. So why do we always start with glycolysis? Because glycolysis is what we would call the central pathway, right? It is the pathway where many of the intermediates can be used by cells to make other stuff, other organic molecules, lipids, fatty acids, purines, pyrimidines, right? Proteins, amino acids. And when you break all those things down, they all can feed back into glycolysis or close to this central pathway of glycolysis, pyruvic acid, Krebs cycle, electron transport chain. Okay. So that's why we keep teaching you this over and over and over again, because it is one of the pathways that almost all organisms use, even if they only use part of it. And it's the one that's in the middle. Like if I brought in a chart of metabolism and unrolled it, that pathway would be in the center because, like I said, a lot of things can feed into it and a lot of the excess can feed out to build other things. So glycolysis, I would consider as sort of like step 1. We're going to go through the complete oxidation of glucose. Remember, oxidation involves loss, and we're talking about not just loss of electrons, but loss of hydrogen atoms, right? C6H12O6. We're going to rip those 12 hydrogens off and end up with carbon dioxide, right? This is also dubbed as aerobic respiration, but only if we're going all the way through to the electron transport chain. Okay, the aerobic part, what does aerobic mean? With what? With air. And particularly not just air, because most air is nitrogen, and that's not going to help us. With oxygen or anaerobic without oxygen, right? So with oxygen, and that oxygen only comes into play because oxygen is the final electron acceptor on the electron transport chain. Okay, so we really literally only call this aerobic respiration when we go all through these 4 steps that we're going to talk about all the way to the end. If something else happens along the way, like we get to pyruvic acid and then we're turned into alcohol or lactic acid, that would be fermentation. Or if we're an organism that lives in the mud or the sludge and we're anaerobic, and oxygen poisons us and kills us as cells, then we're going to do something totally different if we get to pyruvic acid, right? We're going to call that anaerobic respiration without oxygen. So only if we're going with oxygen all the way to the end. So we're going to do that one first, and then we'll talk about the others briefly, okay? So complete oxidation of glucose or aerobic respiration, I would say we've got 4 major enzyme pathways, enzyme steps to discuss here. All right. For each of those pathways, what I want you to know for the test is not the name of every enzyme. Go to biochemistry, learn all about it. Not the name of every intermediate. Don't care. You can just look it up sometime if you need it. These are the things that I want you to know about these 4 steps. Okay. Should probably not put that up there already. I know that's going to make you angry if you've already typed it or written it. Now, I would say this is the minimum you need to know, right? So for the 4 steps of aerobic respiration or complete oxidation of glucose. This is what you need to know. One, you need to be able to tell me to identify the starting and ending molecule for each step. Meaning you need to be able to recognize its name. I won't ask you to draw it or recognize a drawing of it, but you need to recognize its name, and it would be help, and like remember how many carbons it has. Kind of be the limit there, right? Like pyruvate has 3 carbons, acetyl-CoA has 2, glucose has 6, right? So that's one thing you need to be able to tell me for each of the steps. The next thing you need to be able to tell me is you need to keep count. You need to count all the following molecules in each step, okay? How many? The number of ATP that's produced, NADH, and I'll put plus H plus there, produced. How many carbon dioxide are released, and how many FADH2 molecules are produced for each step. And the third thing you need to know that you like absolutely must know is where this happens in the cell. And you need to be able to tell me, and I've left this purposefully generic or vague, where in the cell, right? Both in a prokaryotic cell and in a eukaryotic cell. Right. I am hopeful that this is the third or second, at least second or third time that you've learned this. BIO1, Chem 102. Usually the A&P instructors skip this. It should be deleted from the recording. I don't want to be on record saying that, but I know that usually they skip it. So what we often find is the more times that you learn it, you remember a little bit more of it differently, and you get a little bit bigger picture, right? This is not an easy thing. This is boring. It's awful, right? And you're going to keep learning it over and over as you go on in your studies. If you're not going into nursing, right? You're just going to keep having to do this pathway over and over with more detail. So let's start kind of simple and, you know, see where it takes us. Are they written down? Yeah. Erase. Well, I can go all the way to the sign. So, okay, then now let's start with these 4 major enzyme pathways that are part of aerobic respiration. And the first one is glycolysis. Right. Pretty simple. Glucolysis, right? Lysis to break. It's going to involve adding water molecules or hydrolysis reactions, right? And what are we breaking down? Glucose. So I'm going to do this. I'm going to put C6H12O6 because that's glucose. That's its chemical formula. And then I'm going to draw actually in purple to try to remind you that these are enzymes, and enzymes are proteins, and proteins are always purple. Right? I believe there are 8 or 9 or 10 enzymatic steps, depending on how picky you want to be, to get us to the ending molecule for step 1, which is 2 molecules, not 2 hyphen, right? Not a location of where a phosphate is or a hydroxyl group is, but 2, physically 2 molecules of pyruvic acid. What does your book do? Pyruvic acid, pyruvate. Pyruvic acid, of course you do. Pyruvic acid. Each of those pyruvic acids, so I'm going to put them outside the parentheses, each one of them has 3 carbons. like it's reading my mind, but not quite. All right. So each one of those has 3 carbons. So basically, if I started with 6 carbons and I end up with 2 molecules with 3 carbons each, do you think I've produced any carbon dioxide? Have I lost any carbons? No, right? So all the carbons are still there. What's happened is the glucose molecule has had a phosphate added to it in a couple of places. Literally, it's been snapped in half and rearranged to form these 2 pyruvates. If it's in the form of pyruvic acid, remember acids are proton donors to the solution. So there's a hydrogen on one of the groups. If your textbook or your book is saying pyruvate, that just means the hydrogen isn't there, right? It's already donated it. So kind of like the NADH thing, you can think of pyruvate and pyruvic acid for us. If you can remember it has 3 carbons, that's good enough. Okay. Now, the other molecules that are produced, needed, glycolysis requires an input. Anybody remember of how many? ATP? Yeah, 2. 2 is always a good guess for all of these, just as a, general. If you can't remember, I would guess 2. All right, so 2 ATP, and we'll put those in happy yellow sun, and I'm putting them into the reaction, the set of reactions, right? How many ATP do I produce overall from glycolysis? How many come out of this reaction? Higher? 4. Yep. We produce 4. Right? But we subtract the 2 that we put in. So we say we net 2 ATP from glycolysis because we make 4, right? But we use 2, or we're using 2 the next time we do glycolysis. So we're in, we have up to the positive, we have 2 ATP by doing glycolysis. Acquires 2, makes 4, leaves us with 2. Okay. Now, all of these molecules we've already established we're not making any carbon dioxide, so don't have to worry about that one for this stuff. I can tell you we're not producing any FADH2. Remember, the NADH, its job is to be an electron carrier, and FADH2 is a similar electron carrier to NADH. I'm going to make them blue. We produce, I believe, out of this. Well, that's not a great start to the morning. I believe we produce 2 NADH plus 2 H plus. I'm going to put those in a rectangle to help us find them later. And what's the third thing you have to know for each step? Where in the cell? Anybody have a guess? So we'll do, let's not make it green, let's make it black, because that's important. In prokaryotes, just guess. Not yet, that's a good guess though. Alex can't guess because she can't see. Here, now she'll be able to think clearly. Well, come on, just guess. Where does it happen in eukaryotes? No, all right, never mind, don't guess. Cytoplasm. Yep. And it's the same for bacteria, cytoplasm. Same. So this is the easy one, right? You only have to remember very few things. Starting molecule, ending molecule, how many ATP, how many NADH2, and where it happens in the cell, and it's the same for both. What? Okay, easy peasy, done. Learn that one between now and lab, right? Now, this pyruvic acid is of no use to us because it can't enter into the next step. It can't enter into the Krebs cycle. It has to be changed into something else. Okay. So pyruvic acid is an intermediary or an intermediate that is important in cells because it is the base molecule to build a lot of those things that we talked about earlier, to build fatty acids, to build lipids, to build the base units of most of the amino acids, as well as some of the nucleic acids. But if we are going to completely rip off all the hydrogen molecules from the glucose, which is now in the form of these 2 pyruvic acids, they have to go someplace else. So every textbook is going to show you 1 pyruvic acid. Can you see that, or do I need to write up on? Yeah, because of this, right? Yeah. I was so excited. I went over to the new smart room they're building, and the monitor for the instructor is this big, and it's mounted on the desk. And I went over and I was just like, "How's the room coming?" And they're just like, "See that? That's because of you." I was like, "Yes, yes, the squeaky wheel finally wins." This big, this big. It's like, I'm like, oh my gosh, I'll be able to use so much more of the writing space. All right, so both of these pyruvic acids, not just 1, and it's only one chemical reaction. It's 1 enzyme. So I am purposefully only going to put 1 purple line because it's a single enzymatic step. And these pyruvic acids, each of them are converted into 2 molecules of acetyl coenzyme A. You'll see it abbreviated acetyl-CoA, but it stands for coenzyme. So think back to the first part of chapter 8 when we talked about enzymes as proteins, and they have cofactors, minerals, or coenzymes. Well, here's an example of coenzyme. Really important one. Acetyl groups, you may or may not remember from chemistry, 2 carbons. So now I have 2 molecules that each have 2 carbons. How many carbons did I have in my pyruvic acid? Yeah, I had 3. And so I had 2 molecules with 3 carbons each. So I had a total of 6 over here and 4 over here. How are those released in cells? Carbons. How do we get rid of them? Yeah, carbon dioxide. So I'm going to put it going up like this just because, just because it's a gas, right? Oh, it's going out. Carbon dioxide. I thought that was red. Okay. If you look in your textbook, you will see no ATP are produced or used, but we do produce 1 NADH for each of the acetyl coenzyme A's that are produced. So I'm going to put, coming out of this reaction, 2 more NADH plus 2H plus. Excellent. Where does this happen in the cell? You'll be better at your guesses this time. In prokaryotes? Cytoplasm. Gotta save that cell membrane for, gotta save that for proteins in a membrane that pass electrons at the end. In eukaryotes, this is where we move into the mitochondria. Okay, so in eukaryotes, this happens in the outer membrane of the mitochondria. Literally, oh, maybe I have orange. I don't know why we always draw the mitochondria as orange, but we just do. I'm trying to see where I can put this so that people can see it at home, right? So literally, remember the mitochondria kind of looks like a bean, like a kidney bean shaped thing, right? And the outer, we have an outer membrane and an inner membrane, and the inner membrane is folded up like this, right? It's called the cristae, right? The folds are called the cristae. The outer membrane is still a phospholipid bilayer. So I'm just drawing some phospholipids on there to remind you, as is this inner membrane of phospholipid bilayer. And literally the enzyme that does this reaction, step 2, it's right here. I'm just going to draw it as a purple blob. And I'm going to put a little arrow here from the reaction. So right there is where on the outside is pyruvic acid. As it gets pushed across the membrane, it becomes acetyl coenzyme A. Okay. So it literally happens in the outer membrane of the mitochondria, very specifically. I didn't ask her that. Okay, so I'm going to erase over here. And I'm going to add on step 3 and step 4. So I guess we can do step 3 here. Step 3 goes by many names, so as to help you remember when it's in different books and called different things, right? Your textbook refers to it as the Krebs cycle. It's the only cycle that we're going to talk about in this class. We're not going to do the Calvin cycle, so it's the only one you have to remember. It also goes by the name citric acid cycle. That's what our BIO1 book, that's what Campbell called it, the citric acid cycle. And the reason for that is because the first intermediate that's formed on the cycle is a 6-carbon acid called citric acid, right? So it's identified by the first molecule that's made. Someday, if you go on in biology and or chemistry and you're in a biochemistry class in graduate school, they're going to call it this. They're going to call it the tricarboxylic acid cycle, or the TCA, or tricarboxylic acid cycle, not for the citric acid cycle, although I'm easily confused. And that's because citric acid belongs to a group of organic molecules known as tricarboxylic acids. Okay. Now, do I expect you to remember all of those? No, but for us, anytime you see the word cycle on the test, this is the cycle we're talking about, no matter which of the words I use, right? So the Krebs cycle, technically the starting molecule. So over here, I want you to make a little chart like this. And at the top of it, I want you to write 1 turn of the Krebs cycle, 2 turns, like that. I'll put a line down the middle, so that we can keep track of all the things that we have to keep track of on that list: carbon dioxide, ATP, NADH, FADH2, because all of them are made in the Krebs cycle at different places. Every textbook in the world is only going to show you 1 turn of the Krebs cycle. You have to mentally remember that if I start with glucose, I have 2 pyruvic acids that are going to make 2 acetyl coenzymes A's. So I'm going to have to go around the Krebs cycle how many times? 2 to completely oxidize that glucose. Okay. But every book is just going to show you 1 turn, just like they show you this, usually without the 2's. Right? They're just going to show it to you as a regular reaction. Okay, so here I'm going to write acetyl coenzyme A. I'm going to abbreviate it, and I'm going to draw it like this because it's entering the Krebs cycle. It's joining up with another molecule to form that citric acid. So this is really a purple line. And the first molecule, like I mentioned, is citric acid. So if I ask you what the starting molecule is, it's really citric, it's really acetyl coenzyme A plus oxaloacetate to form citric acid. Then there are many arrows. And we are not going to learn all the names of the arrows. Those are the enzymes. And we're not going to learn the names of all of the intermediates. I don't care, right? Go to biochemistry, learn all about it. So in the center here, I'm going to write Krebs, DCA, citric acid cycle. All righty. So even I have to cheat and look at the Krebs cycle to remember where the NADHs are formed, just to be mildly accurate. Let's see here. It's like at step 3 and step 4. So here, this is really 3. So here, NADH plus an H plus. Here, another one, NADH plus an H plus. At both of those places where we release the NADH, we're also going to let go of a carbon dioxide. So I'm going to put an arrow out for CO2 and put that in a circle again, an arrow out for CO2, put that in a circle again. At the bottom of the cycle, and I know you can't see it very well, but I wanted to leave space for the electron transport chain. At the very bottom of the cycle, we actually make 1 ATP. Happy yellow sun. Yay, energy. Next thing is an FADH2. Put that over here. Comes off this way. Well, let's put him in a triangle. Help us remember he's different from NADH, but sort of similar. And then up here at the top, right before we form that last molecule, I think it's here or here, it's not really important. We make another NADH. Put him back in a rectangle, but no carbon dioxide that time. So if I'm making my little table here, and I'm going to start with the carbon dioxides, how many carbon dioxides do I produce if I go around once? 2. If I go around twice? 4. Very good. How many NADH molecules am I going to produce? 3, he says. And if I go around twice? Ooh, that's a big number. How many how many ATPs do I make? 1 if I go around twice. 2, lovely. And how many of this new molecule do I make? This FADH2. Make 1 of those. So if I go around twice, 2, lovely. Where does this happen in the cell? The colors going here. In prokaryotes, if you had to guess. Cytoplasm, very good. In eukaryotes, we've used up the outer membrane. What is this part called? Anybody remember? The fluid-filled space in between the folds of the inner membrane. Part? No. The folds themselves are called the cristae. What's the fluid-filled space that's like cytoplasm, where the ribosomes are for the mitochondria and the DNA for the mitochondria? Was a famous movie, like in the 90s. Well, Arnold Schwarzenegger in it, I think. Matrix, right? This is the matrix of the mitochondria, and that's where the Krebs cycle is. Yeah, okay. So I'm going to draw it there. And there are more than one Krebs cycle or citric acid cycle in this matrix. The way that the cell makes as much ATP as it needs is because we're multiplying or amplifying this. That's another function of the mitochondria. You know, in 4th grade, we say it's the powerhouse of the cell, right? But this is why it's the powerhouse of the cell, because this is where all the NADHs are produced, that then we're going to use in the electron transport chain to make lots and lots of ATP, right? So in eukaryotes, matrix of the mitochondria. And yes, I wouldn't bother to write that on the board and to talk about it if I didn't think it was important, i.e. that it will show up on the exam somehow, right? Okay, now just briefly, because we're in microbiology, not in BIO1, if I, as the cell, had a lipid to break down, or a fatty, remember lipids, right? Like triglycerides, so they would have a glycerol molecule and 3 long chains of fatty acids, right? So if I was breaking up a fat, saturated or unsaturated, but with those long chains of hydrocarbon, hydrogen with 2 carbons, hydrogen with 2 carbons, hydrogen with 2 carbon, carbon with 2 hydrogens, all the way along, right? I can break, a cell breaks those apart in units of 2, because 2 carbons with lots of hydrogens are very easy to convert into acetyl coenzyme A. This is why fats and lipids store more energy than carbohydrates. If I have a carbohydrate, a polysaccharide that has 6 glucose molecules in it, okay, let's say 12, let's make it a little bigger. Say it's 12 glucose long, it's starch. I just ate some hash browns for breakfast, right? So let's say it has 12 glucose units in it. I'm going to be able to break each of those glucoses into 2 pyruvic acids, all right? So I'm going to go around the Krebs cycle 24 times, which sounds like a lot, right? But if I have the same lipid and it has 3 chains of, let's say, 16 each, and I can break them off in sets of 2, right? Well, let's say it's longer than that. Let's say it's 18. I can get a lot more acetyl coenzyme A's, so I can produce a lot more energy from 1 fat molecule than I can from a polysaccharide, right? For every acetyl coenzyme A molecule produced, you have to go around the Krebs cycle once, right? So if I have a lipid that has, let's say, 45 acetyl CoA units, I got to go around the Krebs cycle 45 times. That's going to make all these numbers times 45, right? That's going to be a lot more than my measly little starch molecule with its 12 glucose molecules. Okay. All right, so the last step. Dun, 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 dun. All right, step number 4. Step number 4 is where we produce all of the energy, most of the ATP, right? It's called the electron transport chain. Plus ATP synthase. Okay, ASE, that means what about this? It's an enzyme. And what do you think it does? What does its name tell you it makes? ATP. Yay! See, I knew it was just the sun in your eyes. All right, so this is the enzyme that makes the ATP. Now, the trick is this enzyme, unlike any other enzyme we know of, this one requires protons, so hydrogen atoms that have had their electrons removed, protons as fuel. And where all those protons are going to come from is here. NADH is going to bring them to the electron transport chain and deliver them so that the electrons are going to go one way, the protons are going to go another way, and the cell can use those protons to make ATP. This part, the electron transport chain, that's where the protons from these hydrogen atoms are going. I'm sorry, that's where the electrons are going, right? And they are going to end up at oxygen, because oxygen's the final electron acceptor on this electron transport chain, because we're talking about aerobic respiration, right? So I'm going to write a bunch of words, and then I'm going to draw something that sort of looks like the picture in your book, right? And we'll go through what happens to the electrons, what happens to the protons. So first, the words. The electron transport chain is 9 or more proteins in a membrane plus ATP synthase. You're like, yeah, that's kind of what you said in the title. Yeah, I'm trying to make you remember this. Duh. Okay, second thing, in aerobic respiration, oxygen is the final electron acceptor on the electron transport chain. That's important enough that it gets its own bullet. All 9+ proteins in the ETC or on the ETC, however you want to describe it, pass electrons. All of them pass electrons down the chain, down the chain. Some, all capital letters, some of the proteins also pump protons across the membrane. So notice I've left this purposefully vague. 9 or more proteins in a membrane, because the electron transport chain happens in bacteria, in the only membrane they have, and it happens in mitochondria, which is the membrane your cells have, and plant cells and protists and fungus. And it also, electron transport chains also happen in photosynthesis, photosystem 2, photosystem 1. Those are also electron transport chains. They're just not electron transport chains with oxygen at the end, because they're producing oxygen, right, and using carbon dioxide. But wait, there's more. NADH plus H plus and FADH2 deliver the hydrogen atoms, I'm going to say, and in parentheses, the protons, H plus to make ATP synthase go, and electrons. I'm going to say to power or to fuel the electron transport chain and ATP synthase. One NADH molecule delivered to the electron transport chain will yield about 3 ATP from ATP synthase. It's an about, it's really 2.86, but you can't have 2.86 children either, right? So we're just gonna round it up to 3. One FADH2 delivers enough potential energy in the form of those protons to yield 2 ATP. That's going to be important for our accounting later. When we're adding up how many ATP do we get from one glucose, we need to know one NADH brings us 3 ATP and one FADH2 brings us 2 ATP. And the last thing on this list, which isn't going to fit, right, is to remind you that ATP synthase, like I said, I've said it, but I haven't written it down, must have protons to produce ATP. You can think of them like fuel. No protons, no ATP. All right, so sadly, that's why I need to be in that room in Madison with the camera that follows me around. So I can write on the board all the way around the room and it follows me. What was that? Yes.
The first step under electric, is it a plus O2?
No, 9 or more proteins in the membrane. It might be 9, it might be 12, depending on the organism. Plus ATP synthase. Yeah, it's a little different than how I presented it in BIO1, right? I'm separating out the oxygen, more highlighting it more importantly, right? Because people aren't paying enough attention to that part. So, yes. Previously, I would have said 9 or more proteins in a membrane plus oxygen, right? And then I would have skipped here to the passing of the electrons, right? But I really want everybody to remember that it's 9 or more proteins, ATP synthase, and oxygen. Because we're going to talk about anaerobic respiration and we're going to take the oxygen out of the equation, but the other 2 things are still there. These 2 things are still there. Okay. Okay, so let's draw a little mock electron transport chain over here. Right, so where in the cell, we'll just add that to our list. In prokaryotes, this is going to be in the cell membrane now because it's the only membrane that they have. And in eukaryotes, it's going to be in the inner membrane of the mitochondria. So here's my phospholipid bilayer, right? Because that's what all membranes are in all cells. Phosphate heads, lipid tails, right? Phosphate heads are charged, hydrophilic. Tails are hydrophobic, right? And embedded in that are groups of proteins. So when you see an electron transport chain, usually you can't actually count all 9 proteins, because some of them are like in this, or like in a bunch like this. And then cytochrome C is always by itself. And then I'm making this kind of shorter on purpose. And then ATP synthase kind of looks like this. It's kind of square at the top, and then it goes like this, and then it's kind of round at the bottom like that. So I'm going to put the membrane in between, and you can draw them however you want, right? It's just to help you remember that it is a physical place and it has to be in a membrane. And it doesn't matter if the cell has mitochondria and chloroplasts or not. There just has to be, these proteins have to be in a membrane somewhere in order for this to function. The first molecule on the electron transport chain, not that this is important for you to remember, but just kind of as an FYI, that first molecule's name is actually, it's an enzyme. It's actually called NADH dehydrogenase. So it's an enzyme that D removes hydrogens from NAD, right? That's what it does. It causes NADH to go back to its oxidized form of just NAD, so it can be reused and recycled inside the cell. So I'm going to bring, let's get some more colors going here. So I'm going to bring this first NADH to the electron transport chain right here. All right. And remember, those NADHs, I've accumulated them in both glycolysis, the Krebs step, and the Krebs cycle, right? From breaking apart the glucose. been a byproduct of breaking apart the glucose. I'm oxidizing the glucose, so I'm reducing NAD to NADH. Remember, those reactions have to be paired. So here comes the first NADH. And remember, I said all of the proteins in the membrane are going to pass electrons, right? So I'm going to make the electrons orange so you can see them. And I'm going to just draw them like this, going along, la, la, la. They're getting passed along the chain. That last protein in aerobic respiration is going to pass the electrons to an oxygen that's waiting here to get those extra electrons. And it's going to become like super negatively charged, that oxygen, because it's getting those extra electrons. That's dangerous. That's a free radical. That causes mutations. In bacteria, it causes death to the cell. In you, just eat some broccoli, right? Has antioxidants in it. That's what that means, right? Helps combat this extra oxygen free radicals that are floating around inside you because you do aerobic respiration. So we're just going to hold on to those electrons and where they ended up for a minute. Okay, it's like they can't do it all at the same time. You can't learn it simultaneously. So the electrons from the NADH are getting passed along the electron transport chain. They end up at oxygen, and now oxygen has extra electrons. Remember, all of the proteins pass electrons. Some also pump hydrogen ions. So let's use a different color for the hydrogen ions, for the protons. Let's make them this red color, this reddish brown color. So the positive charges, a few get pumped here, and they get pumped in pairs across the membrane from the inside to the outside. Some of them are basically being recycled. I think it's two more. And if anyone in Bio1 talked to you about chemiosmosis, right? That's just a fancy word for meaning these protons being pumped back through and producing water, right, as a byproduct. These are the proton fuel. They're all going to go into ATP synthase, which requires protons in order to produce ATP. So the protons are going to be forced through here. They're going to move from one side of the membrane to the other. Two of these protons are going to join up with this oxygen that has extra electrons. Has two extra electrons. Oh, and now there's two positive charges. I can make water. What? Right? H2O. Bloop. Okay, we should have made that blue. All right, I don't have another blue. So it's dead. All right, so I'm making water. Water's harmless. Water's good for other reactions, for lysis reactions. We need water in our cells, right? And it's not that dangerous form of oxygen with the extra electrons. It's just going to come back to haunt us in the next chapter, chapter 7. Well, a couple chapters away. Here, remember what did I say? If I take one NADH electron transport chain, how many ATP do I sort of make? How many? Higher? 3. Good. Right, one NADH gives me about 3 ATP. But what about our other friend here, FADH2? I think I made him orange before, right? That's unfortunate. So let's make the electrons orange. Well, yeah, let's make him pink. Maybe. So FADH2 also comes to the electron transport chain, but guess what? FADH2 is not as good at being reduced from FAD to FADH2. So it doesn't carry as much potential energy. And because of that, because this molecule is an enzyme that has an active site for this isn't close enough to bind there. So FADH2 drops its electrons here and its protons. But notice I've missed the first proton pump of protons out. And I've also missed the first passing of the electrons for the potential energy. So what that means is because I don't have these protons being pumped, they're being pumped here, I have less fuel for the ATP synthase, less potential energy to be converted into ATP, right? So when I bring an FADH2 molecule to the electron transport chain, how many ATP does that give me? Just 2, right? Because ATP, write this down, because FADH2 has less potential energy than NADH. FADH2 has less potential energy than NADH. And I won't try to remind you about the laws of thermodynamics and all that. Just write that down and remember it. Eventually, some point in your life, it might make sense. Or no. Oh no. Yeah. But wait, there's more. All for the low price of $99 a tuition credit hour, right? Here at community college, you can also learn about fermentation and anaerobic respiration. What? All right, but I must rest for a moment. So let's, oh, it's not even 9 o'clock yet. What a great day. All right, so I'm going to share screens with you, maybe, and look at the pictures from your book because they're boring and whatever, but it'll give us both a little rest from you from the writing and me from the talking. So make these big. Okay, now, oh, it turned on all by itself. Pull it down. It's not the one I was on, but okay. Okay, so here is glycolysis. If I can make that one bigger. Apparently, I don't really know how to do this. So I thought there was a button somewhere that I could do that. See, we can take a little nap. Okay. That's weird, but whatever. Just the picture I wanted to say. So enable editing, make it big. There, now you can kind of see it, yeah? Better? A little better? I know your eyes aren't as old and tired as mine, but still. So just as a reminder, I like this picture from your book because for glycolysis, they actually show you the carbons. They're pink, 6 pink little circles. And then again, they're blue. They're using blue for the enzymes themselves. They're naming the intermediates. Again, not important for you. We're going through like this part of glycolysis twice because as soon as we break the glucose in half, one of the halves is ready to go into the pathway. The other half has to have something happen to it. It has to get changed before it can go through the rest of the pathway. And they're keeping them separate and to help you see that you make 2 pyruvic acids. Notice there's just 3 pink balls left down here now, not 6, right? They're showing you the ATP in the happy yellow sun. The NADH is also blue. I've done the same on purpose to help you, right? If you annotate your notes in color and you look at your book, you'll see that it's all the same. The TCA cycle, your textbook has abbreviated to putting the prep step, that step 2, right above the TCA cycle, because in real life, it happens almost simultaneously, okay? So here is what we usually call the prep step. I don't think I labeled it that. I just put step 2, but a lot of times we call it the preparatory step or the oxidation of pyruvate. There's all kinds of chemical names for it. Don't care. On the test in your book, you'll see it called the prep step or the preparatory step. So notice that now we have 3 carbons, right? And now we only have 2, and there's been a carbon dioxide released. And then this molecule is a coenzyme, so it's joining to the enzyme that's grabbing this 4-carbon molecule, adding the 2 to it, and the first molecule that's produced is citric acid. Right? Again, that's why they call it the citric acid cycle. Don't care about all the steps. Just care that you remember 3 NADHs, 1 FADH2, and 1 ATP, and 2 carbon dioxides for 1 turn. That's the reason we made the table. Right? Why do I go around twice? I have 2 acetyl coenzyme A because, just go backwards, right? Because I had 2 pyruvic acids. Why do I have 2 pyruvic acids? Yeah, because 1 glucose makes 2 pyruvic acids. So a lot of times on this chart, I'll tell students to put after the 2 turns if I start with glucose, because you're going around twice if you start with glucose. So probably it'd be a good addendum to your chart. If I started with glucose, got to go around twice. All right. Then I end up with all these NADHs and FADH2s, and I need to take them to the electron transport chain. They've made the proteins different colors, but you'll notice 1, 2, 3, 4, 5, 6. They've put 7 in there. So some of them are 2 or more proteins joined together, like I drew them. It's not important. Here's ATP synthase. So the NADH arrives, drops off its electrons. The electrons get passed. Look, they end up with oxygen. The protons get pumped here. So right away, a little bit further down the chain, a little further down the chain. We're going to use those as fuel. Most of them get recycled. So 2 of them are going to join up with the oxygen to make water. The other 4 are going to just be pushed back into this space below the membrane to be pumped out again. They're just going to be, let's keep going. FADH2, notice coming to electron transport chain late. Do you want to think of it like that? Not as much potential energy, so not as many protons are pumped, so you get less ATP. Yeah, I'm going to make a different table, and then we'll talk about anaerobic respiration. Let's stop that share. Questions you have about this so far before I erase and proceed? Yes, no? Do you want to photograph it or you just want to go find it on the video? It's totally up to you. Do you want to take a picture of it? Go ahead. Now's your chance. Faster at that. All right. We're in cell. We did that. So I don't think we missed anything. Did we miss anything? Yes, no? No? Good? All right. So let's get rid of all this. And let's talk about what if we are not doing aerobic respiration? What if we're doing anaerobic respiration? Should have left my membrane up there. All right, so let's try to use it. As I mentioned, pretty much almost all organisms are are capable of glycolysis. Some version of it. There are modifications, but again, go to biochemistry, learn all about it. And I would remind you that at the end of glycolysis, we end up with these two pyruvic acids. All right, put those in a circle. And basically there are three types of pathways that pyruvic acid can go down, generally, if we're breaking it down for energy, not using it to build something. And one of those is aerobic respiration, which we just talked about. And the high points of this to remember are oxygen is the final electron acceptor, because it's aerobic respiration. Now, you are going to produce, so I'm just going to write this, Krebs cycle present. And if we're in a bacteria, you are going to be able to produce a maximum of 38 ATP per glucose. That has to be in yellow. Happy yellow suns per glucose. Now, these other two pathways, these other two general pathways, are going to produce less ATP, but they're not as involved. They don't have as many steps, they don't have as many enzymes. And if I'm a bacteria and I'm just a single cell, I can still live this way. All right. So I'm going to talk about anaerobic respiration next. Oxygen is not the final electron acceptor, something other than oxygen. It's usually another inorganic molecule is the final electron acceptor. And what we mean by that, it's usually a molecule that does contain oxygen, but it's not oxygen by itself. So it's usually a nitrogen, like a nitrate, or a nitrite, or a sulfate, or a sulfite. It's usually a nitrogen or a sulfur molecule derivative of some sort. Doesn't have to be, but often it is. The Krebs cycle is present, but it's usually abbreviated. So I'm going to put short Krebs cycle, incomplete Krebs cycle. And remember, that's where we make all the NADH. All right, we made 3 for 1 turn, 6 for 2 turns. We're taking all those NADH to the electron transport chain to make those 38 ATP. Because we don't produce as many NADHs, we're not going to have as much fuel for ATP synthase. So we're not going to make 38 ATP from 1 glucose. We're going to make in the neighborhood of 20 plus ATP per glucose. I can't tell you how many because it's different depending on the bacteria, and it's also different depending on the conditions the bacteria are in at the moment. So remember when we talked about inclusions in chapter 4, and I told you there are things like sulfur granules or nitrogen deposits, and there are things the bacteria needs for its metabolism. And then we wrote that down and we started talking about endospores, right? That's what I meant. So if I'm a bacteria that does anaerobic respiration, I have to have a store of sulfur in order to make my ATP, right? If I'm living high off the hog and I'm living someplace where there's lots of sulfur, good for me. If the environment has just changed dramatically, or the temperature has changed, or my conditions have changed because I live in the soil and my soil got moved, right? Maybe now I'm not making so many ATP. Maybe now I'm only making 15, not 28. All right, so it varies, depends on the bacteria and the conditions that bacteria is in. Okay. The third choice is fermentation. And it's really important that you understand, especially in microbiology, that fermentation and anaerobic respiration are not the same thing. Because bacteria that ferment, they can do it in the presence or absence of oxygen because they're not using the oxygen. It doesn't matter. Okay, so it is a little bit of a misnomer when your anatomy and physiology professors say you do aerobic respiration in your muscles until you run out of oxygen, and then you start fermenting or doing anaerobic respiration. You're fermenting. You're not doing anaerobic respiration because there's still oxygen there, just not as much as there was 4 minutes ago when you started lifting those weights. Okay, if there was no oxygen there, you'd be dead. So that's, but we use it like a different way in different, like subtopics. Fermentation, an organic molecule, is the final electron acceptor. And I'm just going to put, it's usually an organic acid, like lactic acid, succinic acid, could be alcohol, right? If we're yeast. There is no Krebs cycle, lack Krebs cycle. So if there's no Krebs cycle, I'm not making extra NADHs to take to an electron transport chain, which doesn't exist. Right? So there's no Krebs cycle. Oh, I forgot on this guy. There's also, not only is a short Krebs cycle or partial, there's also a short or partial electron transport chain, ETC, also short. These guys don't have an electron transport chain. So they're only going to produce 2 glucose per ATP. Sorry. Maybe I need more coffee today. 2 ATP per glucose. But I'm a yeast cell, or I'm a single bacterial cell, and I don't have to do all those other enzymatic reactions, so I don't have to build all those other proteins. So I can do this pretty fast. Like just have to do glycolysis in like 1 or 2 steps, and I get 2 ATP. And do glycolysis, couple steps, get 2 ATP. Right? I don't have to do glycolysis, prep step, Krebs cycle, electron transport chain, move it around from the cytoplasm into the mitochondria, or the cytoplasm to the membrane. Right? So I can do this a lot faster. And if I'm just a single cell, I can live like this. Now, what we're going to see in the lab is that there are bacteria that do combinations of this. They have the equipment to do aerobic respiration, but similar to your cells, if there's no oxygen available, they'll just start fermenting, and they won't multiply as quickly, but they'll still survive and multiply. Okay, so we'll see that some organisms can do both. Some organisms do this, anaerobic respiration and fermentation. The last thing we're going to do before we go to lab is to make a summary table, because I forgot to do that before, so that you can count up where all those ATP come from. Because I suspect if you've learned this in anatomy and physiology, or even in BIO1, usually we abbreviate this number and say you don't actually get 38, you get between 32 and 34, depending on the cell you're in, and how, because it costs ATP to move the intermediates to the mitochondria, and then it costs ATP to get them out of the mitochondria and into the rest of the cell, the ATP. So since we're in bacteria and we don't have mitochondria, we're going to go with 38 because it makes for better accounting, in my opinion. So I'm going to erase this, and we're going to do a quick summary table, and then we'll go to lab. All right, so summary of the complete oxidation of glucose. So if we start with glycolysis, over here, I make a little table, and we put ATP and NADH and carbon dioxide and FADH2, right, like that. And I say, we start with glycolysis. How many ATP do we produce from glycolysis directly? We're doing net. 2. How many NADHs do we produce from glycolysis? What did I tell you was a good guess for everything? 2, good. How many carbon dioxides, except here, how many carbon dioxides are produced just from glycolysis? The notes that you just took. 0. How many FADH are produced from glycolysis? Just from glycolysis, nothing. That was just from glycolysis from step 1, right? What is on an out arrow coming out of the reactions? Okay, on that prep step, the turning the pyruvate into acetyl-CoA, do I make any ATP directly on that step? Were there any happy yellow suns on that purple line? No. Did I make any NADH? 2 of them, because I had 2 pyruvates that became 2 acetyl-CoAs. Did I release any carbon dioxide? 2. Did I? It went up. Remember, it was in that reddish brown. And any FADH2s? None. Excellent. Now, if I go to the Krebs cycle, TCA, citric acid cycle. How many ATP do I make? I got to go around twice because I'm oxidizing glucose. So how many ATP do I make directly from the Krebs cycle going around twice? 2, good. How many NADH do I produce? 6, lovely. How many carbon dioxides do I release? 4, good. And how many FADH2s do I make? 2. How many ATP do I just have? If I gather these up, just the ATP I already made. 4. Okay, so put those there, save them for later. How many NADH do I have all together? 10. Where do they go? It's okay, you can say the words. Where do they go? Electron transport chain, good. And ATP synthase, right? That's weird little things, right? Hanging down. How many ATP? If I take 1 NADH, the electron transport chain, how many ATP do I make? 3. So if I take 10, how many will I make? 30. Wow, that's a lot. What about my friend FADH2? How many of those do I have? So if I take 2 FADH2s to the electron transport chain, how many ATP will they make? 4. I'm going to make them in pink so you can remember they're from the FADH2. Now, how many ATP do I have all together now? 34 plus 4. Yes, I have 38 ATP altogether. If I'm doing aerobic respiration and I'm in a bacteria, right? Not in your cells, but we're not talking about humans in this class, right? Except as host, right? And being able to defend against bacteria, but we're not talking about how you make energy. Chapter 8 was called Microbial Metabolism, right? So all 38. And if you're a bacteria, that's a lot. That's from 1 glucose molecule you've transported in. If you also happen to transport in some lactose and some fructose and some galactose, and you can break those down and put them into glycolysis, right? You've got hundreds of ATP, which you can then use to build protein, to make enzymes.